This is what we call a starch print, and we've been able to print S202 because of the distribution of starch in the cells of the leaf. If you like, the leaf acts in a similar way to a photographic print, and the image is formed because of the different distribution of starch within the cells. To do this, we first of all kept the leaf in the dark for some time so that its starch reserves were depleted, and then attached an exposed photographic negative to one of the leaves and let it in the light for a few hours. The negative acts like a mask, and where light passes through the clear part of the negative, then photosynthesis occurs and starch, a reserve carbohydrate product, is formed. To make the starch visible, we put the leaf in boiling alcohol, and this dissolved out the chlorophyll, and then the leaf was put into iodine, which stains starch blue. So this shows in a dramatic sort of way the sensitivity of photosynthesis to light levels, but it's sensitive to other factors too. Temperature, for instance, varies enormously in different habitats, and so does the availability of water. This can indirectly affect photosynthesis because of its influence on the opening and closing of stomata through which gas exchange occurs. So plants in these very different environments are adapted physiologically, that is, in their mechanisms of photosynthesis. What we need to know is exactly which factors affect the rate of photosynthesis. So we need to be able to measure the rate of photosynthesis. In principle, measuring the rate of photosynthesis is a simple matter. A leaf is enclosed in an illuminated chamber, a stream of air passed over it, and the rate of flow and concentration of CO2 in the air is measured as it enters and leaves the chamber. By subtraction, we can calculate quite easily the net amount of CO2 taken up. In practice, it's a bit more difficult than this because the stomatal pores on the leaf surface through which the CO2 gets into the leaf are also sensitive to the factors we've been looking at, temperature and light intensity and CO2 level. But you've therefore got to control these factors. Well, now here, the jacket round the leaf chamber controls the leaf temperature, and this thermocouple actually monitors the temperature at the surface of the leaf. The temperature of the air coming in is controlled by this water bath here, and the humidity is also controlled in the same way. And the input airflow and the output airflow are analyzed for their gas content with these infrared gas analyzers. This analyzer tells us the difference in CO2 level between the input and the output air. So at any one time, you can look at it and see what's happening. But if you want to see what's happening over a period of time, then it's all displayed on this chart recorder. Now here, in steady conditions, we've got leaf, or rather air humidity, leaf temperature, and CO2 levels. Well, let's go back to the beginning of this experiment. We're going back in time now. And I lift out the chart to do this. There we are. Here's the start. Now, at the beginning, this straight line, there was no CO2 difference between input and output from the chamber. When the lights were turned on, when the experiment was started, then gradually CO2 uptake began. And this is net CO2 uptake because, of course, the leaf is carrying on respiring whilst it's in the chamber. Now, a bit later on, we varied other factors. Here, for instance, we varied temperature. Temperature was raised, and very shortly afterwards, the rate of photosynthesis, the amount of CO2 uptake, was increased. And here again, you see, temperature up, CO2 uptake increased. So it's very sensitive. Even a very short time after you vary a factor like temperature, rate of photosynthesis changes. So we can vary the factors with an apparatus like this. In fact, with something like CO2, we kept it constant. And up here, we record that the level of CO2 in the air was about 330 parts per million, which is approximately what it is in the outside air. Now, in the field, CO2 and all the other variables can change a lot. And if it's a very hot, still, sunny day, CO2 levels around the leaf can fall very low, and they soon become rate limiting for photosynthesis. In fact, in the commercial production of tomatoes, it's common practice to enrich the air in the greenhouses with extra CO2 to raise productivity. So, in the field, it is important to be able to monitor these factors when they are all changing and varying. And although this apparatus is absolutely splendid in the lab, it isn't exactly portable. Out in the field, we can use a different technique, even in the middle of winter. This time, we use radioactive CO2 to label the products of photosynthesis. And to do this, part of a leaf is clamped so that it's enclosed in a gas-tight chamber. A pulse of radioactively labeled CO2 is then fed into the chamber. During the labeling period, when the 14CO2 is being taken up by the leaf, light intensity, leaf temperature, air temperature, and relative humidity in the chamber are all measured and recorded. The whole process lasts about a minute. The area of the leaf that was enclosed in the chamber is then cut out and killed instantly by dropping it into liquid nitrogen. So back in the lab, the leaf segments can be analyzed for 14C, and we get a measure that's close to the rate of gross photosynthesis in terms of amount of 14C fixed in a unit time per unit leaf area. Now, if we look at a subtropical crop like maize, there's a marked response to temperature. It doesn't usually photosynthesize below 10 degrees centigrade, but the rate rises steeply up to almost 40 degrees before falling away. In a temperate species like ryegrass, however, photosynthesis occurs even at 0 degrees, rising to a maximum around only 23 degrees centigrade. Compare ryegrass to maize, and you can see how poorly ryegrass performs at high temperatures. Not surprisingly, responses like this can lead to big differences in plant growth. It was the German Nobel Prize winner Otto Warburg who provided the first clue to explain these differences. Warburg showed that in the green alga chlorella, the rate of carbon dioxide fixation, and thus photosynthesis, was markedly affected by oxygen, especially at high temperatures. Photosynthesis was highest at oxygen concentrations far below those of the atmosphere. Indeed, the rate declined progressively as oxygen levels increased. This decline was accompanied by secretion into the medium of a 2-carbon compound, glycolic acid. A similar effect was found in higher plants, like ryegrass. But here, glycolic acid is found in the leaves. The second clue relates to this blip on the CO2 recording from the gas analyzer, the erga. Now, remembering that the midline here indicates there's no net efflux or influx of CO2, if we go back to the beginning of the chart, when the leaf was put into the chamber, first of all, in the light, there is an uptake of CO2. It attains a steady state of photosynthesis. But here we switch the light off, and of course photosynthesis stops. There's an immediate drop. We pass the midline, so we're now looking at something like respiration. The rate of respiration tails off until back here there's only a very low basal rate of respiration. Now, this big blip is an indicator of a special kind of respiration that only goes on in the light. It's called photorespiration. And what we're seeing is a kind of carryover from the light for a brief period in the dark. Of course, the rate of photorespiration must affect the rate of net photosynthesis. And photorespiration is sensitive to the same kinds of environmental factors as photosynthesis. Look, for instance, at what happens if we raise the temperature. Now, the temperature here of the leaf is 27 degrees instead of 17. Here, the light was switched off. and look at the size of the blip. There's an enormous rate of photorespiration. So photorespiration is increased at higher temperatures. It turns out that the substrate for photorespiration is glycolic acid, which you might remember from before is the substance produced when oxygen inhibits photosynthesis. And indeed, if we look at the effects of lowering oxygen level, now here, light off, oxygen level is now 2% instead of atmospheric 20%, and there is virtually no blip. Here's the midline, virtually no blip. The same kind of thing happens if you raise the level of CO2 but keep the oxygen level high. So what seems to be happening is some kind of competitive interaction between oxygen and CO2, and this links up with the rates of photorespiration and photosynthesis. The basis for this interaction is a chemical one. Let's consider what normally happens in the fixation of CO2. It combines with the 5-carbon sugar ribulose bisphosphate, RuBP, and the reaction is catalyzed by the enzyme RuBP carboxylase. The product is a 3-carbon compound phosphoglyceric acid, or PGA. This is the first intermediate of the Calvin cycle, and the chain of reactions that follow lead to the net formation of sugars and regeneration of RuBP. Now, RuBP carboxylase can also combine with oxygen. So oxygen and CO2 compete as substrates for a site on the enzyme, and at high oxygen levels, oxygen fixation becomes the dominant reaction. It leads to the formation of glycolic acid, which is the substrate for photorespiration. This involves some release of CO2, so there's a net loss of carbon, but eventually 3/4 of the carbon from RuBP is recycled to PGA and re-enters the Calvin cycle. So CO2 fixation and the Calvin cycle are strongly inhibited by oxygen fixation. But through photorespiration, at least some of the carbon from glycolic acid is salvaged. Clearly, though, oxygen competition presents problems for the plant. It's in deserts that the problems of photorespiration are likely to be most severe. Here in Death Valley, California, it can be extremely hot and dry. In the day, plants close their stomata, and although this prevents water loss, it also restricts carbon dioxide uptake. So you'd expect photosynthesis in these plants to be affected by low levels of CO2, with strong inhibition through oxygen competition and photorespiration. But in this cactus, a typical succulent, if we measure photosynthesis by the rate of formation of sugars, we find that it's not affected by oxygen levels. The rate stays more or less constant. And at high temperatures, there's no inhibition. It continues to synthesize sugars when many plants would have wilted. So how do they do it? These succulents have an ingenious system whereby they open their stomatal pores only at night to fix CO2. And of course, water losses at this time are much lower than in the day. When they fix the CO2, it is fixed into a 4-carbon acid, malate, and this is stored in the cell vacuole. And the next day, CO2 is re-released from the malate and fixed into the Calvin cycle proper. And because all the re-releasing goes on in the chloroplast, the CO2 is concentrated and the problem of photorespiration is overcome. This kind of metabolism is called crassulacean acid metabolism, after the family Crassulaceae, where it was first found. So what we've got here is a system where the initial and the conventional fixation of CO2 are separated in time. But the price the plant has to pay is that first, it takes more energy to do it this way, and secondly, the plant is strictly limited by the amount of malate that it can store in its vacuole. And this probably explains why a lot of these succulents, especially the cacti, are such slow growers. But other plants have been found in hot tropical climates that have exceptionally high growth rates. They too have solved the problem of photorespiration. They're called C4 species because, like CAM plants, they fix CO2 initially into a 4-carbon acid. But unlike CAM, this is not restricted to nighttime. This is a C4 crop. It's sugarcane, which is a species of grass, and this particular crop's in Hawaii. The plants here are about over 3 meters tall, So it's a pretty hefty grass species. And the sugar, sucrose, is in the stem of the plant, the cane here. And if you look at the leaves of this plant, you get the typical C4 pattern. If you hold them up to the light, then the veins stand out not as pale, but as dark green because of the Krantz anatomy. That's the bundles round the veins, which are filled with chloroplasts. In this cross-section through a sugarcane leaf, you can see chloroplasts concentrated in the cells around the bundles, forming a bright green wreath. This is why it's called Krantz anatomy, Krantz being German for wreath. If we look at a chloroplast from these bundle sheath cells, they look very different from normal. They have parallel lamellae, but stacks of grana are conspicuously absent. In C4 plants, only these chloroplasts contain enzymes of the Calvin cycle. The mesophyll chloroplasts have a more typical structure. There are grana, and it's here that oxidation of most of the water takes place with the release of oxygen. But these chloroplasts contain no ruby P carboxylase. CO2 is fixed by a cytoplasmic enzyme into a 4-carbon acid, and the acid must be transported to the bundle sheath cells for re-release of CO2 into the Calvin cycle. Here are chloroplasts from the mesophyll and the bundle sheath side by side. The bundle sheath chloroplast is much larger. The dark areas are starch grains. Lack of grana here means that no oxygen is produced by the light reactions, which helps to reduce photorespiration. But in addition, entry of CO2 into the Calvin cycle is concentrated here by the breakdown of the C4 acid. So in C4, in contrast to CAM plants, the initial and the conventional fixation of CO2 are separated not in time, but in space. The effect, though, of re-releasing all the CO2 in the bundle sheath is to concentrate it and therefore also to overcome the photorespiration effect. So C4 plants are not usually inhibited by oxygen and they're rarely restricted by CO2. In both kinds of plants, though, they're very good, they're very efficient at fixing CO2 and removing virtually all the CO2 from the air, and also at refixing any that's released in either photorespiration when it does occur or in conventional respiration. Now this means that they have very low CO2 compensation points, the point at which CO2 efflux and influx are balanced. And I can demonstrate this with a simple experiment. In these two flasks a few hours ago, I closed a leaf of a C4 species and a C3 species. And inside we put a little bit of universal indicator in these small flasks. Now that indicator will absorb CO2 from the air, and when it does so it goes red. And what you see is that the C3 species has indeed got a red indicator. So this species, when it came to equilibrium, when it reached its compensation point, had quite a lot of CO2 left in the flask, whereas the C4 species had virtually none. It had removed nearly all the CO2 from the inside of that flask, and the indicator didn't change colour. So there's this much lower CO2 compensation point in the C4 species, and what this means is that they can maintain very steep gradients of CO2 from the outside air into the leaf. That means the CO2 keeps diffusing in more rapidly. Even when the stomata are partially closed, this happens. So this is one of the reasons why C4 plants make very efficient use of their available water and still photosynthesize at the same rate as a C3 species. Compared with C3 species then, you expect C4 plants to do particularly well in very hot conditions, bright sun, good tropical conditions, and often rather dry. On the whole, they don't do particularly well in the shade. But one exception that's known to this is a species related to this species of euphorbia here. Now, the species I'm talking about occurs only in Hawaii, and it grows in the shade. It's a real shade plant. Even more surprisingly, though, is the discovery of C4 plants in the British Isles. These were discovered by Professor Woolhouse and his group, now at the John Innes Institute. And among them is the salt marsh grass, Spartina.
We turned to salt marshes for this purpose to screen plants because the highest productivities are found in salt marsh communities in Britain. And one of my students quickly found 3 species of C4 plants in British salt marshes. One of them, this plant, Spartina, was particularly interesting because it's a dominant over thousands of acres of salt marsh around the coasts of Britain.
To find a C4 plant growing in water and in a cool climate like ours is most unexpected, so it was important to prove conclusively that Spartina had a C4 mechanism. Fresh sections of Spartina examined under the microscope showed that there was indeed evidence of Cramp's anatomy. In this case, there's only half a ring of cells with dense chloroplasts. Further confirmation was obtained using another technique. Sections of the leaf were put into a solution containing antibodies of the enzyme ruby P carboxylase, and then mixed with a fluorescent dye that binds to the antibodies. Under the microscope, the bright yellow-green of the dye shows up only around the bundles. So it's only these cells that contain ruby P carboxylase, as you'd expect in a typical C4 plant. Could it be then that the C4 mechanism has advantages that may usefully be introduced into other plants in this country?
Now, the problem for Britain is that at least in our pastures in the southern and eastern parts of the country, we know that water is in short supply and depresses grass growth and hence dairy yields in the summer months. And so we must ask the question whether if we could introduce the C4 mechanism into pasture grasses with its greater efficiency in the conservation of water, whether this would enable us to maintain grass yields under these conditions. And this is part of our experimental program at the present time. It poses problems for understanding how the C4 mechanism is controlled genetically, and then how it might be introduced into species of our temperate grasslands.